All right, everybody, we're going to go ahead and get started. Thanks, everyone, for being here today at our research seminar. My name is Cinnamon Moffett. I am the Research Program Manager at Oregon State University's Hatfield Marine Science Center, and I will be your host for today. For those folks that haven't been here before, and I'm looking around and I recognize most of you, but I'll say it anyway, this is a hybrid event, so that means we have folks online and folks in the room. For folks that are online, if you have any questions for today's speaker, or if you have any technical issues, our volunteer Roseanne, who's in the room with us, will help you navigate those technical questions, and we'll read out the questions to today's speaker. You just need to put them into the chat box. But please note that your cameras, mics, and screen shares have been disabled for this event. For folks in the room, because this is a hybrid event, when we get to the question and answer section, you will need a microphone. So if you just raise your hand, you get to watch me run around and get you a mic so you can answer those questions. I also just want to remind folks, if you're here for credit, make sure you've signed in. Dr. Stafford is not able to be here today, so visually she doesn't know you're actually here. So make sure you sign in so that she can keep track of that. I believe today's your last class. Woohoo! So nice job, everybody. Thanks, Lisa. All right, a couple of quick announcements before we get on with today's presentation. Wanted to let you know that next week on December 14th, we have our next seminar. Cheryl Barnes is from the Integrated Marine Fisheries Lab, which is part of the Coastal Oregon Marine Experimental Station, also known as COOMS, here at Oregon State University's Hatfield Marine Science Center. She's going to be talking about navigating uncertainty through stakeholder engagement in fisheries. It is also our very last seminar of 2023. So it's been a good year, everybody. So hopefully you can come to that last seminar of the year. If you want to know more about any of our events, you can go to our homepage, scroll to the bottom, and there's a calendar of events there. And you can get all the information you need to either join us online or in the room. But you're all here today to learn about sharks and not listen to Cinnamon talk. So just want to do a quick introduction of today's speaker. So today we have James Sulikowski, who's the director of COOMS here at Oregon State University's Hatfield Marine Science Center and the Astoria Seafood Lab. He joined the Marine Science Center here in May of 2023 after being an associate director at the School of Mathematics and Natural Sciences at Arizona State University. Dr. Sulikowski has a bachelor's degree in biology from Denison University, 2 master's degrees in marine biology, and 1 in physiology, and a PhD in zoology from the University of New Hampshire. So we are excited to learn a little bit more about Dr. Sulikowski's work. He focuses on sharks, rays, and skates, studying their movement patterns, growth rates, and their resilience to climate change and fisheries pressures. And that's what he's going to talk to us about today. So I'm going to hand this off to James.
Okay, can everybody hear me okay? Yeah, all right. Yes, we're going to talk a little bit about sharks, but apex mothers, right? The cool part of what we do is to focus on a very specific life history stage. So a little bit about what we kind of do here. So I'm going to be pushing a ton of buttons on this, and I'm going to make a mistake for sure. So just kind of bear with me on all that. Okay. All right. So part of the Big Fish Lab here, Taylor and I sort of integrate ourselves together, which is super cool because he does a certain component of biology, more of the behavioral component of it, and I do a little bit of the ecophysiology of it, the reproduction. And together with our movement overlap, it's kind of a perfect circle of shark biology. But I'm today going to be talking about sharks as moms. I know everyone's excited as I am about this because I love this. And so there's lots of funny things in this. Feel free to laugh. Awkward silence is never good. I will say that there are some scenes of dissections and things like that. I'll forewarn you on those as well. So if you want to turn your head on those. So what do we do here? Well, reproduction takes a couple of individuals here. And one of the things about sharks is that they have internal fertilization. Yes, they do. And this is these are claspers here. And this is a male. This is how you look and define a male. If you didn't know that, you go in an aquarium, you see one of these sharks swimming by, you can be like super smart and be like, that's a male, right? And people will be like, oh my gosh, how do you know this? Well, you know, cell presentation. Anyhow, there is part of the internal fertilization for sharks is that they have to reproduce, they have to mate, okay? And you might want to turn your head on this one. There's a lot of graphic stuff going on here, but basically, the male has to pin down the female. And so there's an act of sort of aggression. There you go, they're mating. And there's this clasper, okay? And he's inserting it into the female here. But you see, she's covered in bite wounds, right? And females actually develop thicker skin during this time to adapt to that. But it's all part of that process. And once we've got that, you know, internal fertilization, this is where it gets really interesting for us in our sort of science. Now, if you look inside a shark, they have a very unique kind of reproductive set of organs. They have a uterus, which is this right here. And then they have ovaries, which has the follicles, which has the eggs. Those eggs get ovulated, and then they go through this structure right here, which is called the shell gland or navimental gland. And all these sort of structures work together to produce the new set of offspring. So how does this all kind of work? Well, once mating has taken place, that sperm actually travels all the way up this uterus, goes to the shell gland here, and is stored. It can be stored for up to a year. It's pretty remarkable. And the next phase of that is when that reproductive cycle starts, then that follicle is ovulated, goes through that, the ostium into the shell gland, gets fertilized, and the next step is for an egg case to be created. And as that follicle is falling through that shell gland, this gets created, and depending on the reproductive mode, would depend on how thick that egg case is. Now, this is where it gets really interesting for us, is that there's actually 5 different reproductive modes for sharks. The most simplest is oviparous, which is an egg layer, or you can have viviparity, or sort of live birth. And there's a bunch of different modes under that. And so I'm going to walk kind of through all those because it's super exciting for us. Egg lengths, just as it seems, just like a chicken, except they don't sit on them, okay? They hatch and they kind of swim away. If you are a placental viviparity, the mom basically becomes the egg case. It dissolves inside of her. There's a yolk. The yolk is used for nutrition. Baby get born alive and they swim away, not backwards, but they swim away. Okay, I was too lazy to change that. Then you have oophagy, okay, where mom, after the yolks are gone, mom keeps creating eggs that are ovulated, and the babies eat it, and then they're born and they swim off. Then you have intrauterine cannibalism, which is amazing. How would you like that? Basically, in this form, the yolks are dissolved. Mom stops creating eggs for them to eat, and then they start to eat each other. And the first one to develop teeth basically eats everyone else in the womb. So mom gives birth to 2 babies, one in each uterus. And then finally, we have placental. And that's the most advanced form. And it's just like a mammal. There's a placental attachment, deriving nutrition from that. Basically, after baby reaches maturation, it's born, swims off, and they start the cycle over again. So how do we know about all this? It's pretty amazing, right? We got this information from gross dissections, okay? And this is one of those images. Learn a lot from that. However, nowadays, we're running into some issues such as conservation, right? It's really hard to dissect a individual that is threatened or endangered, right? And if you look at the IUCN Red List, about 75% of elastomeric shark stays and rays either fall into data deficient, right? Or they fall into a status of near threatened, vulnerable, or even endangered. So we have to find a way to do this type of work without sacrificing them. In addition, elasmobranchs ranks are typically late maturing. They have slow growth. They produce few young and live really long. They're a lot like us. So dissecting them, killing them to find this information doesn't really work in their life history stages, but also they're subjected to a bunch of other sort of insults, such as overfishing, climate change. So all these factors really kind of play into needing to find a different way of doing this. And so for us, when we talk about conservation, understanding where these animals are is really important. What's their critical habitats, but also reproduction, okay? When are their cycles? When do they reach maturity? And if we can connect these two, we can learn a lot about where moms go, okay? This is a mom, she's on her way. This is a bull shark. So we want to know where mom goes. But again, if this is what you have to do to mom, you can't figure out where she goes. So how can you do this a little bit differently? Well, it can start with the brain, believe it or not. And sharks have not very well-developed brains, but they do produce a couple of key hormones that we can study, okay? And just like us, you have a gonadotropin-releasing hormone, and you have gonadotropin, and basically those impact the reproductive organs. And here we're talking about the ovary. And from that, okay, that organ, we can get different types of reproductive hormones produced. Testosterone, estradiol, progesterone, the same ones reproduce. And we can actually take a blood sample from a shark or stingray or other species. And instead of kind of doing this, okay, we can do this, or we can actually take a muscle biopsy. And the animal can live, which is great because that hormone concentration then can tell us if the animal is pregnant or not pregnant, okay? And the nice thing about that is that mom can then go on her way. She can swim by. That's great. So the problem with hormones is that you got to wait, okay? And everyone asks, is it pictures of the dogs going to be in this one? Yes, it is. That's Bosco and Zoe. If I leave for 5 minutes, this is what happens, okay? And it's the same thing with hormones. You have to wait for them to be analyzed, right? So we don't know if mom is pregnant or not pregnant, if the shark's sexually mature or not, until a couple of days in the lab. And so I have some students, Kiki, Lauren, and Kirsty, that are learning those techniques now. And they'll learn that painful process of how long it actually takes before you get a result, okay? So we need different ways of doing this. And one of the things that we work with is an ultrasound. And you can see here, this is a Dr. Shark performing one. There's actually a clinic down the road where this happens. And we can actually see this at the point of care, the point of contact. And what's great about that is that instead of this to find, you know, out if it's pregnant or not, right? We can use an ultrasound. And this ultrasound then allows us to link movement to reproduction on the spot, which is great for us. And the cool thing is that what we see inside here is pretty amazing. Those are baby sharks that we see in our ultrasound. So it's like kind of like Christmas when we see this, or a happy holiday that we get to see these amazing things. And that's the eyeball right there. So we can take these on the boat. We can do this, and then what we can figure out is what stage she's in, which is amazing. And this ultrasound works just like any ultrasound, right? At a vet's office, at a doctor's office. It's the same technology that is used on humans, except ours is veterinary grade, right? And it's mobile, and it's waterproof, it's water resistant, it's durable. We can take it on the boat. And so I'll show a larger video here. And so this is, here's the gills, it's a backbone, it's a mouth opening, closing, that's an eye. So it's super cool, right? First times we've seen these, everyone was totally blown away of that's a baby shark, that mom's pregnant. Now, the other cool thing about this is that we can measure the size of the embryo, right? And so that'll tell us at what stage of gestation that shark is. And for those that have been dissected, or we know a size at birth, we can then extrapolate out how long her gestation is going to be and where she might give birth, which is really important when you talk about elucidating habitats that are needed for the survival of those species. So how do you link reproductive biology to movement then? Like, how do we actually do this? Well, we can use satellite tags, all right, to track them. And they're super cool. We use two types. The top one here is a fin mount tag, and the one on the base of the fin is called a pop-off satellite tag. They both kind of do different things. The fin mount tag is awesome in that when it's above the water surface, okay, there's a wet-dry sensor. And when that wet-dry sensor senses that it's dry, it transmits a location. Okay, so it's off when it's underwater and it's on when it's out. It's pretty amazing, huh? Worked all night on that. Okay, so that gives us a real-time location, right? That fins out, transmits. We know where that shark is. It's really cool. The problem with this one is that the shark has to come, its fin has to go out of the water. And only a few species actually do that. So how would we kind of measure that in a species that doesn't do that? Well, we can use this pop-off satellite tag, okay, which collects temperature, depth, and also geolocation from light levels. And this one's great because it archives the data, okay? Once it's underwater, it just starts recording. It records for as long as we want it to. If it goes to the surface, it doesn't transmit. So after, we'll say, a year, it'll pop off, and then it'll dump all that data to a passing satellite. And then we put it together like a piece of a puzzle. And again, my students are going to be learning that painful process. But it's pretty amazing when you can get those results. So how does all this sort of come together in real time, right? And so this is some of our work in New England on porbeagle sharks. You can see that there's the ultrasound. There's somebody getting blood in the back. We're looking, oh yeah, she's pregnant. Amazing. When the shark's out of the water, it's got water going over the gills so it can breathe, like us going underwater with scuba gear, which is pretty awesome. Okay. Once we know she's pregnant, right, then the team sort of assembles, right? And we start putting on the satellite tags. And it's like a NASCAR event. Everybody's working together to kind of do their thing. And it's amazing to see, and it happens so fast that when you're done, you're like, what just happened, right? But the after effects of that are super cool, right? That final push, when you actually push the shark overboard, okay, and you see her swim away, and you can see the tags on her, you know that she's pregnant, and you want to watch where she goes, which is super cool for us. And porbeagle sharks are related to makos and white sharks and salmon sharks. It's actually a salmon shark of the East Coast. But if you put a mako and a white shark together, squish them together, that's basically a porbeagle. They're super cool. So I'll give you an idea. This is a track of 3 of our females. Each color is a different female. And this represents animals that have been swimming around for about 9 months. Okay, and so we want to know where they go and where are they at certain times of the year. So you can imagine we tag them right around in here. Okay, and this is their movement. This is in October, and you can imagine this is all their movement the rest of the year. And they start heading back here in May, right? And what we want to know is what spots here are they giving birth? Okay, and if they are giving birth here, right? And we've narrowed it down, okay, to this area, right? So just a little spot, right, where birth may be taking place. So, you know, for us, it's a little frustrating. And how do we know that? Well, we get little baby porbeagles in these areas. Okay, they're so cute, right? We call them pork chops because they're super cute, right? But if I don't know if you can tell, I'll put a little pointer on here. This one actually has a little satellite tag on it. And so we want to know where the babies go as well. And this tag actually, it's a kind of a prototype, but it's meant to grow with the shark so that it doesn't damage the fin or anything like that as it starts to mature. So how do we put these pieces of the puzzle together? And this is the frustrating part for us in that, you know, there's mama sort of swimming around, okay? And where does she actually give birth? right? Is it there, right? Is it there? Is it there? Okay, and again, that's a big spot, right? This area right in here. And these little suckers are highly mobile, right? A little porbeagle can go 100 miles in a day, no problem, right? They can just beeline it. So determining these areas are really important for us. The reason that we find really interesting in this species is that as soon as you cross this international sort of boundary line, right, it becomes an endangered species in Canada, right? It can actually be harvested here in the United States. So it interacts with the tuna fishery in the United States. There's discard issues in Canada. So we want to figure out ways in which we can figure out, how do we work with the industry so that we can reduce the interactions with these sharks in certain areas at certain times of the year. And this is why, we're really interested in figuring out where these certain life history stages are going at certain times of the year. But again, it's frustrating because these are big swaths of space. And who knows, maybe these little sharks were given birth offshore and swam hundreds of miles upshore, inshore to these spots. So we developed a tag to figure that out, to find the exact timing and location of birth. We got so frustrated, we said, all right, we're just going to do it. And this tag is coined the birth tag, and it's basically looks like an egg, okay? And the way that this tag works is that you stick it in mom, and it stays in mom next to her babies, okay, throughout gestation. And then when she gives birth, this tag pops out with the babies, floats to the surface, and transmits the location, okay? This works a lot like the fin mount tag. It's got a wet dry sensor on it. And so when it's in mom, it's wet. It's in a sense in standby mode. When she gives birth, comes out, floats to the surface, transmits the location. As soon as it hits the surface, because it senses it's dry, right? So that was our concepts behind all of this. And I'll walk through this a little bit. And so this is it in action, okay? This is actually a tiger shark upside down. There's the applicator. It's going inside of her. And, it's just a 12-foot tiger shark. It's no big deal, right? She's in tonic immobility. It's all good, okay? So once that tag is inside of her, we know she's pregnant, so we've done the ultrasound, okay? She's pregnant. She's got a fin mount tag on her, or even a PSAT, all right? And then we let her go, right? And you can see her fin mount tag. She swims off. She's like, what just happened, right? I can get out of here. I've been abducted by aliens. It's crazy. So that shark, actually, we did a lot of work with tiger sharks in Bahamas because there's a big dive operation there. It's called Tiger Beach. There's lots of tiger sharks there. We want to know about the reproduction. Was this area serving as a nursery habitat? So we tagged this animal, and she was amazing. She transmitted for close to 3 months in this area, and then all of a sudden she just disappeared. Right? We don't know what happened. Was she caught by a fisherman? Did the tag stop working? What happened? What was up with the birth tag? We didn't know. We thought all was lost. And then on May 20th, the birth tag actually popped up, which was super cool for us because it popped up in a known tiger shark nursery ground. And for us, that was just like, oh my God, I couldn't believe this. It's like, literally was like, woke up and it's like Christmas morning. It's like, I can't believe this, calling everybody. This is incredible. And so it was like our first step to this, that it actually worked. And this area was, prime baby tiger shark area. If you look at where the size of this animal when it was captured, the babies were about 30 centimeters in total length. They're born at about 60, 70, right? And so we analyze this against what we know about potential gestation rates and size at birth and all these things. And this made complete sense to us, that what we observed was an actual birth tag going the distance and being deposited here in this nursery ground. So that was really exciting for us. What wasn't exciting for us was that we had about 5 years of failures of this tag throughout the process. We had firmware issues, we had battery issues, we had antenna issues. So that shark that stopped transmitting, and all of a sudden the birth tag popped up, there were 10 other sharks that were tagged that the birth tag didn't work because we found out there were firmware issues. We thought we had a firmware issue fix. Go 10 more sharks, guess what? There's a battery issue. And then we found out there's an antenna issue. They can only wiggle that much before it doesn't wiggle anymore, which was tough. So we did a lot of testing for the next 2 years to figure all those kind of things out. So we started this process in 2017, and finally this March, it kind of all came together, which is amazing. And one of the shark species that we did a lot of this testing on was a sand tiger shark. And this was a sand tiger shark we filmed, or we got filmed doing, but it was pretty amazing because this was the teeth. This shark, when we did the ultrasound on her and released her, was literally going to be born any day. This thing was like 105 centimeters long. It was incredible. So it was like perfect for us to test if these tags were working. Put it on a shark right before it's going to give birth, pops up, great, it's working. We did that on 2 or 3 other species to make sure that it worked. Prolonged deployments, a week, 10 days, 2 weeks. Finally, it's working. So it was pretty exciting for us. But what these shows don't show you is some of the behind the scenes stuff. And so this is actually the release of that shark. Okay, that's us trying to push it off the boat, right? It's got stuck. It's trying to bite people. You know, I'm trying to get overboard. It tries to bite me. You know, and we release it, and it goes swims away. But for us, it's really exciting because it's carrying this next generation of technology to hopefully kind of get us to the next step. And so that next step came, you know, a few months, basically, and we tagged a hammerhead shark in March of this year. And this was sort of the deciding fact that all the upgrades have been made. That's the hammerhead here, and you can see how big she is. That's her head. That's her tail. That's us performing the ultrasound on her. And the cool thing here is that that's what a baby hammerhead looks like, right? That's the eyes. That's the mouth. Looks like E.T., right? It's super cool. You know what I mean? Everyone says, that looks like E.T. So it's pretty awesome. And of course, when you are going to put technology like this in the shark, you have to make sure she's safe and whatnot. You can see there's the host is giving her air or water over her gills. We take that out. We release her. She swims away. And then everyone's like, oh my gosh, what just happened? This is, you know, totally incredible experience for everybody. And of course, it's rough out and everybody's kind of half sick. And it's just an amazing kind of incredible experience for us. What was most amazing about this shark, right, was this process right here. And so she was one of the most incredible transmitters of a location that we've ever had. So that's her track. She was at the surface probably 30 or 40 times a day, right off North Carolina. And you can see that's where she spent all of her time. And so we were watching her and waiting for her to do something. And all of a sudden, she makes a beeline, right, for South Carolina. In short, birth tag pops off and she leaves. Okay, so it was really incredible to see all this stuff kind of come together for once, right? Which is pretty awesome for us too, is that we actually went to South Carolina and tried to catch baby hammerheads. And we did catch baby hammerheads. And that's what a baby hammerhead looks like. They're so cute, aren't they? They're amazing. Yeah, they're pretty awesome. Okay, I like to say jawsome, you know, but there you go. But it's a tough life down there, right? That's a little baby hammerhead that was eaten by other little baby sharks. I mean, they have no shame, and they bite. Okay, that's my hand. Okay, and then you can see a little baby hammerhead bite right here. So you're never safe. So what do you do? What do we do here, right? So we've been all over the place doing this technology, but we're really excited to do it here. And there's a lot of really cool species to do this. You've got white sharks, mako, salmon, seven gills, blue sharks, the list goes on. And so we've actually done it on some blue sharks here, and I'll show you a couple of tracks. And blue sharks are pretty amazing species. And here's some that were swimming around one day on the water. I mean, look at them all, right? They're all over the boat, and we literally know nothing about their movement patterns here or how they fit in the ecosystem in Oregon. So we're like, this is a great opportunity. So what are we going to do? All right, well, let's do some ultrasounds, see if one's pregnant. And yeah, it was pregnant. It was pretty cool. So what do? You know, we give them the whole 9 yards, right? Put a tag on them, put a birth tag in her, kick her overboard, and then we wait. We see what happens. I can tell this shark has been a really incredible transmitter as well. Blue sharks actually really do well on the East Coast. We didn't know if it did the same on the West Coast, but that's her track. She was tagged in August, okay? And you can see her kind of moving around, and then slowly migrating south, okay? And just this morning or yesterday, I can't remember what it was, she's pinged off Hawaii, right? So this is why these tags are so cool. You can really get a good idea of these secret lives of where these animals really are, okay? So put a tag on the outside, put a tag on the inside, know she's pregnant. We can really start to understand critical habitats, migratory pathways, linkages, right? These animals don't care about boundaries. I mean, this shark may end up over, who knows, you know, Australia, Japan, who, you know, it's heading that way. So we're really putting the important touches on really understanding these sharks in a different way right now. But, you know, there's a lot of sharks out there that are just too big, right, to study, right? Like, how are you going to catch this, right? What are you going to do with these types of sharks? You know, basking sharks, whale sharks, like these are sharks that are critically endangered globally, right? How do we help them and get the information that they need as far as reproduction? Well, you can't catch them, but you can swim with them. So we've developed an underwater ultrasound, right, to swim by a shark to see if she's pregnant, okay? Now, the cool thing is that, you know, we can work with dive operations, others, to see how those sharks are being impacted by ecotourism helping them. So there's a lot of really cool questions that we can answer with this, and that's a bull shark that's kind of swimming by, and these sharks were pregnant. And so it adds an extra layer of sort of conservation to these groups. So being able to develop this technology is really important for the advancement, right, of conservation and management. And I'll show you this, which is super cool. This is a hammerhead, which was done in Bimini. And this shark had been coming to this location for, I think, 9 years. And all the dive operators, you know, had named her. Name was Gaia, strong connection to it, okay, and really wanted to know how they were impacting those species. And so we took the ultrasound there, and we did all this. And you're going to have to wait till Shark Week this year to find out if she was pregnant or not. So I'm sorry. So that's a little bit about what we do, okay, in my lab. Someone was worried that I was going to be over time, I could tell, okay, with all the slides, but I did it. Just want to say thanks. I'm excited to be here. There's a lot of great people, a lot of great research, and I feel like there's a lot we can do and accomplish. So Mama Shark says thank you, and always happy to discuss more about the research and how we can collaborate and so on and so forth. So that's it.
Scared you into ending early,
did.
so that's great.
Yeah.
That means we have a little bit of question time. Do we have any questions online, Roseanne? Okay, for folks online, if you put your questions into the chat box, we'll work through those together. How about questions in the room? All right, hang on.
Nice talk, James. I'm curious, You talked about females. I'm curious about the males and if you know about, or how you're disentangling where they mate. And then how many do you usually have to catch to get all the pregnant females that you showed us? I imagine it's not as easy as you.
Yeah. Oof. Yeah, that's a great question, right? Because every time we talk, everybody like, oh, you just go and catch them. It's super easy, right? So no problem, right? It took us about 8 years to find out where the pregnant females were in the Port Regals in New England. That was working with the commercial fishing industry and whatnot. And we've got a better understanding of kind of where there's aggregating at certain times of the year. So we can go, typically in the fall and do pretty well, right? Spend like 4 or 5 days and probably catch 30 sharks and maybe 8 will be pregnant. So not bad, right? We've gone at certain times of the year and caught 1 shark, right, in 5 days. So it's super frustrating. You just never know. In the Bahamas, I'd say about 30% of the sharks are pregnant that we catch. They seem to be like asynchronous in the way that they're kind of reproducing. So we're focusing really hard on females right now, just because, look, they're the ones that are sort of carrying the next generation, right? And looking at, how can we understand their corridors? But the mating is really important as well. We have tagged a couple of males, and their patterns are so different. It's really cool, right? Like all of our females are off the Cape Cod and kind of going out in maybe a little south. The males are all going north, like into Canada, which is really cool. So detangling that would probably be the next step. Like we really want to know where mating kind of takes place. We know about when, but where is also really important to protect those areas, obviously, as well. Yeah.
All right, we have a question online. Go ahead, Roseanne.
Do each of the 5 modes of birth correspond at all to the shark species?
Yes, I would say you have families, right? So part of the way the taxonomy has worked for the different families, right, in sharks is the way that they reproduce. So for example, all the egg layers, this family, all the ones that give live birth with placental viviparity into a family. So yeah, you can group them like that.
All right, questions in the room. You all are going to maybe run back and forth on one side to the other.
Great talk, James. It's kind of a follow-up to that question. Are those patterns of the reproductive mode borne out by the phylogenetics?
Yes. Okay.
Yeah,
I'm trying to think of it's complicated in a sense. Reproduction is sort of like a fallback, I would say, in all the phylogenetic taxonomic things that we've been working on, right? I mean, you have weirdos, right, in this, like these groups, but for the most part, it's pretty standard.
All right, Roseanne, go ahead.
Were the birth tags used for oviparous and viviparous?
That's another really good question. We have not used them in an oviparous species, only viviparous. Only viviparous. Part of that is because of the gestation or the amount of time that egg case would stay in a female. Usually an egg case only stays in a female about 24 hours, that process. So maybe a little more complicated to do that with them.
So James, I know you probably only show us the really good videos, but I was amazed at how docile these creatures seem. I mean, there's no way you could handle a Chinook like you do. Is that a true assessment of it, or is there any use of some ways of anesthetizing them, or?
I'll show you something. real quick. I'll give you an idea. So I took, actually I need to take it out of slideshow mode. I've showed you the real calm sharks, and I'll show you this one. Okay, this is that's a tiger shark that was too big.
Hang on, you
Oops,
stop
oh,
sharing.
sorry.
Hang on just a second.
Okay.
All right, we have some technical difficulties. Hang on, everybody. You want to answer another question while I pull up your slides again?
I'll show you, Michael. You'll see. So I'll verbally tell you that they are not calm. Okay, that they are absolutely. It takes us a long time to sort of get them to the boat, calm them down on the boat or beside the boat. Yeah. So it's a little bit of a challenge for us. And so this is a tiger shark that's was too strong for us, right? And you can see it just pull the buoy, that buoy's about like this, like right away. And they get a little nuts, but that's okay. But yeah, it's challenging for sure. Usually it's like somebody sitting on a shark, somebody holding the tail, somebody holding the head, right, to kind of restrain them. Yeah.
All right, go ahead, Roseanne.
Do you think it will be possible to study deep water sharks the same way?
Yes, we are hoping to be able to do that. One of the things about deep sea sharks is that they don't have a swim bladder. So a lot of the pressure issues that most other fish deal with, they don't. But the tags that we're using are good, I'd say deep sea to a certain degree, right? They're rated to about 2,000 meters. So we can study sharks in that realm right now. So we're working on tags that can go deeper. It won't get crushed by the pressure, but yeah, I think so. And that's an area that we're really interested in actually, because we know zero about those groups.
All right, James, can you go back into your blue box while we ask another question? Thank you.
My blue box.
So that was really great. Thank you for sharing all of that lovely data and video with us. I was wondering if you just got lucky catching those baby hammerhead sharks off of South Carolina, or you knew where to go, you went directly to where, the tag popped up, or if it's known to be a nursery.
Mm-hmm. Yeah, really good question. So, when you're developing a tag like this, you want to make sure that, look, what's happening is real, right? It's not just like you put it in the shark and it came out too early or did something like that. So we want to work with species that we knew where the nursery grounds were, right? And tiger sharks, hammerheads have really well-defined nursery grounds. And we work with groups all along the coast. And when this thing popped out, we actually, called our friends in South Carolina, like, oh my gosh, it's popped out. And so we went down and we sampled with them. And they've been doing the work there for years. I'm like, oh yeah, that sounds exactly where, it should be, which is super cool. And what's really cool is that there's a cryptic species of hammerhead in the Carolinas. It's called the Carolina hammerhead. It looks exactly like a scalloped hammerhead, but they're going to different places to give birth, right? And because this one went where it went, they're like scalloped hammerhead. Yeah, which was super cool. Yeah. So those are kind of things that come out of this research that I find really exciting, kind of interesting.
All right, we'll take a couple more questions. Roseanne, looks like you have a question online.
Thank you so much for sharing your work. Have you found that different species in the same geographic area have the same seasonality when they're pregnant?
That's a great question. So I would say that one of the most complicated things about what we do is that what we think we know, we don't know, right? And we're finding a lot of plasticity in shark reproduction. And you can have a shark, a species that will seem to be an annual reproductive mode, right? And then you'll catch another one that's showing signs of biannual, like skipping a year. And it's really interesting to us how plastic all these reproductive modes are, and what does it mean? And those 5 modes, the reproductive hormones do about the same thing, but they all do a little bit different things within the mode. So that's another level of complexity. I mean, we see, you know, females that are mature, and you'd think they'd all be synchronous in the reproductive style, but there's a group within there that's biannual, that's pregnant, and then you have a group that's biannual and not pregnant. So it's really complicated. And the more we dig into it, the more it's like, oh my gosh, there's a lot more we need to be looking at. It's not linear, like what we think in reproduction.
Nice. I think there was a question over here. Okay. I got a couple of them.
Thanks for that. I'm curious about the application of the multiple kinds of tags on the same animal. So I think you showed mostly the fin tags, but and I was curious what kind of data you're collecting from the pop-up tags, and if you have been able to match, or if you plan to be able to match that up with the birth tags to be able to look for behaviors that would actually indicate the release of the pups, and without actually having to use the birth tag in the future and, you know, the invasive tag.
Yeah, I'd say the 2 parts of that is that the fin mount tag gives you, it's only for certain species that come to the surface, which is great, right? And it's real-time data, it's amazing. But it can't archive the data constantly, right? And so we use the PSAT, we typically double tag, so we can get a temperature and a depth, so you can kind of a 3-dimensional spatial idea of where that animal is when it's not at the surface, or it's collecting additional information. So the 2 of those tags together give us a really good idea of where those animals are, what temperature preferences, what depth preferences, and define the habitats a little bit better, right? So that we can understand what their range is and what their needs are. Okay, so that's one aspect of it. The birth tag was developed because for pelagic species, the babies are really mobile, right? And do they give birth inshore? Does a mom come inshore like a hammerhead or whatnot, or they're offshore and the babies move inshore? And so that was why that tag was developed. We can kind of guesstimate when birth will take place, right, from the ultrasound images. But, you know, there's so much variability within size at birth, timing and whatnot. We can probably get it down to like a couple of weeks, a month maybe, when that might occur. And so then it raises the question, is where exactly is it happening, and how do we protect those areas, right? And how are they shifting and whatnot? So I think the birth tag, the idea, the concept was to take it to the next level, to the next step, so we could tell exactly where this was happening, so that we'd have a better idea. And if it was offshore, right, what commercial fishing, what's going on in those areas that we could identify and maybe protect at that particular time. Right, and so that's kind of where we were with this. That answer the question?
If you only concentrated on one species with a specific question on movement behavior, what would it be?
Well, if I had $1 million, right? I mean, here's the thing. Honestly, like we're interested in the question, not so much the species, right? All the species are super cool for us. Everyone's got their favorite species, right? Mine's the port beagle. I love the port beagle. But we really want to work through and compare, right? Like a mako here on the West Coast to a mako on the East Coast. What's the habitat preferences for those species, right? They're the same species, but they're in different geographic locations. Is the timing the same? Are the preferences the same? What's the evolutionary stamp? You know, what does that mean evolutionarily? So that's where we are sort of headed, or I'd like to head with the research, is asking those more broader questions, right? And how does the same species life history moms differ in different geographic locations? So that's how I would pursue it.
So now that you're here on the West Coast, have you considered working on salmon sharks? And I ask this somewhat selfishly. I'm with ODFW across the parking lot, and I get about a dozen calls every summer that a baby great white shark is washed up on the beach.
Yeah.
One of them got transported to Idaho as a prank and showed up on a river in Idaho. But there's some thoughts, there's pupping grounds off of Northern California somewhere. This gets picked up by social media, the news stations out of Portland, and gets a lot of attention once a year. And I know that's something that a lot of people in Oregon would be really interested in, more information on the salmon sharks or the baby great whites that are on the beach.
Yeah, that's a high interest species for us for sure. As the salmon shark, just from multiple different aspects of it. I mean, just because it's the porbeagle of the West Coast, and they're literally identical, right? I mean, down to the double caudal keel on their tail. But yeah, we're really interested in that species. We just can't find them. We would love to, right? It's just really hard. There's not a lot of baseline information for us to go figure out where these animals are and to be able to afford to go find them and fish for them and catch them and release them. And we get a lot of anecdotal data from fishermen, but you know how that goes, right? It can be challenging. But yeah, salmon sharks are like top of the list for sure.
Roseanne, any questions online? Okay.
Thank you for sharing. I have a question about, so basically as humans and all of the different detrimental factors we've impeded on all other inhabitants of the earth, I think we kind of have a responsibility to do conservation work to take care of these species. But at what point do these other anthropogenic like trauma that is being introduced to these species, at what point is the research worth the, I guess, ethicality of it, if that makes sense?
Yeah, totally. So I can tell you this, each one of those sharks, if we put a sat tag on its fin, we put a PSAT on a pop off and a birth tag in it, that's like $5,000, right, in that animal. So all of our work has been to ensure the survival of mom, right? And also, make sure that nobody gets hurt in the process. So for us, understanding how those animals are utilizing their environment and how we can reduce our impacts on them, right? So you can imagine that there's an urbanization area, right? I don't know, somewhere along the coast. Lots of people in the water, love it, right? All of a sudden climate change comes in, and that's the new pristine habitat for shark species. They need that area, right? So if we know that that is going to become that area, right, we can work in order to ensure that we can do things to allow that shark, that mom, to have access to it. And so the things that we do are all geared to ensure that whatever's happening to mom, right, is not going to hurt mom or the babies. So all of our techniques are approved by veterinarians, and you can go down the huge list, right? And so we're really observant of that fact, right? And so we don't want to hurt mom. And look, anytime you catch a shark, it changes the behavior for a little bit, but everything that we've seen, all of our tags are meant to fall off, right, after a certain amount of time. So mom's not carrying anymore. And so we've kind of worked ways in which to ensure that that doesn't happen to hurt her.
Okay. Any last comments from you that you'd like to share with us before we wrap up?
Thanks for coming. Yeah.
All right, everyone. We're going to wrap it up there. Thanks so much for being here. One more round of applause for today's speaker. All right, everybody, go enjoy while we're not getting rain right now. And hopefully we'll see you next week at our last seminar of the year. Thanks, everyone. Thanks, everyone online.
Hi. Yeah.
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 Over at ODF.
Yeah. How are you?
Good. So I I guess I might follow up with Lynn's question.
Yeah.
She asked you about salmon sharks.
Yeah.
but, You know, another West Coast species, you know, spiny dogfish.
Yeah,
Right?
yeah,
And
yeah.
I think there's a suspected pumpkin ground.
Yeah.
off of
Oh,
the
man.
Nearshore waters of Washington.
Mhm.
I'm just wondering if you guys were thinking.
We are,
Okay, for folks online,
yeah,
we're gonna wrap
totally.
up now.
So we're
Hope to see
working
you
with
next week.
Leaf, yeah, on some spiny dogfish stuff.
Very cool.
But-
Very cool.
But